It's time for This Week in Science and Education, the internet show that talks about all things health research and science related, new discoveries, new cures. How do we take this information and how do we make it relevant for the teachers and students in today's classroom? We're going to find out next. Stay tuned for This Week in Science and Education. This Week in Science and Education is brought to you by Sheridan Institute of Technology and Advanced Learning. At Sheridan, students shine brighter. Visit their website at SheridanShineBrighter.com. Hey everybody, it's time for This Week in Science and Education. This is episode 45, recorded Tuesday, June the 7th. Paths to Cancer Research Careers. Joining me today is Dr. James Koropatnick. Jim, thanks for joining us today on the show. We really appreciate you stopping by. Dr. Koropatnick is a researcher here at the University of Western Ontario, but actually holds his home at the hospital. Jim, tell us a little bit about that and what you do.
Okay, well, I'm at the Cancer Research Laboratory program at the Lawson Health Research Institute. Now, the Lawson is the research arm of the London Health Sciences Centre. And the London Health Sciences Centre is the main hospital in London, Ontario, along with St. Joseph's Hospital, Parkwood Hospital, and a whole family of hospitals that tend to work together. But the Lawson is that research arm that takes care of all research among those multiple hospitals. So I do my teaching, undergraduate teaching and graduate teaching at the University of Western Ontario, and I'm a professor at the University of Western Ontario. But all of the graduate students that I get, of course, are from the University of Western Ontario, and they're working in my laboratory in the hospital system at the Lawson Health Research Institute at the hospitals. So my laboratory, along with multiple other laboratories that are part of the Cancer Research Laboratory program, are all housed in a Victoria Research Building, which is right next door to the Cancer Centre in London, Ontario. So there's a large number of patients being treated in the Cancer Centre right next door to the Cancer Research Laboratory program. That's very important because the graduate students in my laboratory, Christine DiCresce here and Saman Maleki Vareki, and other graduate students in the laboratory are right next door to the clinicians who are actually treating patients. So one of the great strengths of the research program that we have is that we're spending a great deal of time talking to our academic clinical oncologist colleagues who are treating patients while we're working in the laboratory. So they come into the laboratory to work with us, and we often get into the clinic to actually see what's going on with the patients. And that's both a huge challenge for us, but also extremely exciting, because it's not very often that we get to work with our clinical colleagues face-to-face and hand-in-hand all the time. Very difficult to do on the main University of Western Ontario campus, but a wonderful place to do it in the hospital system.
I want to get into that a little bit, Jim, if we can, but first, why don't we actually introduce your graduate students that you brought along with you today and tell us a little bit about them and what they do.
I'll start, and then they'll introduce themselves a bit because they know what they're doing even better than I do, because I should say, just to start, that in my laboratory we have a number of graduate students and a number of postdoctoral fellows and research associates and clinicians, but the people who actually do the work at the bench, and they do all the work that actually ends up being published in journals, and they do all the work that we hope is ultimately going to end up as a better treatment for cancer patients. That's all done by the students, and it's all done by the research associates, and all done by the technicians. So they do the actual work. So they're going to tell you about what goes on at the bench. But first of all, I'll introduce Christine DiCresce. And Christine is a PhD candidate student at the University of Western Ontario in the Microbiology and Immunology Department. So the university takes care of that graduate training. But in my laboratory, she actually is the one working at the bench in that hospital setting. And she'll even tell you a little bit about some of the work she's doing with clinicians in the hospital system as well. And she's particularly interested in overcoming cancer treatment resistance, which may be a bit of a mystery to some of the people who may be listening, but Christine will tell you what it's about.
And do you mean from a psychological perspective of patients resisting treatment?
No, the tumors themselves.
Okay, okay.
So strangely, tumors have the capacity to learn from being exposed to certain kinds of treatments.
Oh, no,
and when they learn from that, they can learn to avoid those treatments. So Christine is particularly interested in trying to overcome the capacity of cancer cells to resist treatment.
Okay.
And Saman Maleki Vareki is a PhD student in my laboratory as well. And Saman is particularly interested in the immune system that exists in patients' bodies. And now it turns out that our own immune systems have an inherent capacity to recognize cancer cells and to destroy them. And there are ways for cancer cells to learn from being exposed to the immune system so that they avoid being killed by the immune system. And when they avoid being killed by the immune system, they have certain tricks to do that. So Saman is devoted to the idea of overcoming those tricks that cancer cells play on the immune system so that the immune system is able to uncover and unmask cancer cells, find them and kill them. So he's trying to tune up our own immune systems so that they're able to kill cancer cells endogenously, perhaps even without external cancer treatments or drugs. So Saman will tell you just a little bit about that.
Sounds great.
Okay, yeah, thanks Jim for the introduction. I joined Jim's lab with an immunology background, and what I'm interested into is the interaction between the immune system and cancer. As Jim mentioned before, cancer cells can fight back immune system. It's called cancer fight back. And what I'm trying to do is to enhance the immune response, and by looking or downregulating an enzyme which is called IDO, which is released or produced by cancer cells, and that enzyme can suppress the immune system. Now, I'm trying to look at the interaction between the immune cells and those cancer cells that produce this enzyme, and to see if with downregulating that enzyme I can induce the immune system. And because immune cells have different ways to kill cancer cells, and if we can just help these cells and just induce the response a bit, they can overcome hopefully this allergic state and then kill the tumor cells in general.
So give us a little idea, because our audience, of course, is high school students 9 to 12, and give us an idea of the steps you would take in order to ascertain such things. How do you conduct your research, I guess, is the question.
To give you an idea about what I do is, I got these cancer cell lines that we obtained them originally from patients. These are cell lines that they have obtained from patients, real cancer patients, and now we have these cell lines growing for years in the laboratories, and we work on those cell lines. Now what I'm doing is I treat these cancer cell lines with a chemical which is called interferon gamma, which I call it the ultimate gun of the immune system. And what that chemical does is to induce IDO expression, which is that dangerous enzyme, by the cancer cells. Now I'm trying to, after inducing that enzyme in those cancer cells, to downregulate that enzyme by treating the cells with siRNA or with small molecules that they go inside the cell and can bind the mRNA or RNA inside the cell, inside the cancer cells, and downregulate the messenger response to produce IDO. And then we are hoping that to make these tumor cells more susceptible to the immune system by treating cells with siRNA.
It's an interesting process Saman has just mentioned, because he's talking about using human cancer cells that perhaps years ago came from cancer patients. And when they came from those cancer patients years ago, we immortalized those cells, which means that cells that come from patients will grow for a period of time outside the body, but they often die away after a while. It's hard to maintain them outside the body. These are cancer cells that have become immortalized. They've escaped that death that occurs outside the body, and will continue to grow for years and years and years. Well, there's a whole lot of processes that go on in those cells that make that happen. It has to do with some genetic changes that occur in those cells. Their chromosomes are no longer degraded the way the normal chromosomes are degraded, and they may have turned off some genes that are normally suppress growth and survival, and they've turned on some genes that make the cell survival continue. But in any case, they go for a long period of time, and that allows us to explore things outside the body. So in the laboratory, there's several steps that we want to go through if we want to move from discovery phase in the lab and move this all the way to treatment of patients. The first one is to try to make discoveries in the laboratory to try to find out, are there certain molecules in cells that we can target to either turn them on or stop them from being made, like IDO, as Saman has mentioned, that will ultimately have a chance of being targets in real patients later on. So once we make those discoveries, let's say we establish that IDO is a molecule that if we can turn that molecule off or stop it from being made in cancer cells, it'll make cancer cells susceptible to the immune system. We can do that perhaps entirely outside the body. Then we'll go to what is usually an animal model. So we may go to, in the case of IDO, treating mice with this same molecule, but very special kinds of mice. These are mice that have to have partially a human immune system. So in fact, there are mice that have been generated that actually express certain portions of the human immune cells, and we can grow human cancer cells in those mice as foreign cells. And then in the mice, we can watch the human immune system attack the human cancer cells. And then in that mouse system, we can begin to treat the mice with molecules that can stop cancer cells from making IDO and ask, does that make their tumors shrink, or does it stop their tumors from growing? So this is partially between an entirely non-human, non-animal system and part way to the human system. Then if we can get good results from that kind of an animal experiment, next stage is the human studies. And that's the biggest leap that we have to make. But luckily, we're in a hospital system where patients are being treated all the time. And we have clinical colleagues who are anxious to take things from the laboratory and into what are called clinical trials. That's where drugs or new treatments are actually tested in cancer patients. So once we've identified, for example, that IDO may be a target that we want to stop tumor cells from making in human patients, we will make the molecules that can do that. Those are the new drugs that we want to test. With our clinical colleagues, we'll do all the appropriate, what's called preclinical testing, to make sure that those drugs are likely to be safe in cancer patients, as safe as they can possibly be. Then, with our clinical colleagues, we'll go to what are called phase 1 clinical trials, and we will very cautiously test those drugs in cancer patients at very low doses. And then when we find a good, safe dose to treat patients with, then we'll go to phase 2 trials. Phase 2 trials are those where we want to ask, are these treatments actually doing any good? Are they actually helping to treat the cancer, stopping it from growing, or perhaps getting rid of it entirely? So we're looking for a response in the tumor. And if you get to that phase, then you go to the phase 3 trial, which is what's called the make or break trial. That's where you ask, is this new treatment better than the treatment we already have for those cancer patients? And if it is, and you can prove that to the health regulatory agencies around the world, then it becomes a standard of care for cancer patients later on. Now, I have to say the biggest gulf here is between the laboratory and the clinic. How do you get to that phase 1 trial? And in fact, we do have some drugs now not targeting IDO, but targeting a different molecule that Christine DiCresce is working on. And we are beginning to move to phase 1 trials with that particular drug. And the reason we're doing it and capable of doing it is that we're right in the hospital working with our clinical colleagues. And Christine, maybe you want to talk about that particular target.
You know, it's really important to have a good backup these days. We all have information that we want to protect, and we want to make sure that we can retrieve. There's a lot of stories, and I'm much a culprit as anybody on this, that we just don't have adequate protection anymore on our computers. We seem to be so busy with our lives that we don't take the time to sit down and set up an appropriate backup. Documents, pictures, research, all sorts of things that we have on our computers, and we've got to make sure that it's protected. Well, how are we going to do that? Why don't you give Mueller Systems a try? Their website is muellerdata.com/VROC. That's muellerdata.com/VROC. If you visit that website, free for 90 days, you can download, install, and configure their software. Make sure that it works for you. If you get stuck or you're not sure how to do it, give them a call. They'll walk you through it. Again, that website is muellerdata.com/VROC. We thank them very much for their support of our podcasting programs at Virtual Researcher on Call, and we look forward to working with them some more in the future. muellerdata.com/VROC. Get that data protected, folks.
So project that I work on focuses primarily on an enzyme called thymidylate synthase. And
give that word again.
It's thymidylate synthase.
Okay.
TS.
TS,
Yeah, we'll shorten
okay.
it to TS. So as a population of cancer researchers, we've had TS as a target in cancer therapy for quite a long time now. It was, previously developed in the 1950s to target cancer with chemotherapeutics.
Oh.
And my project is using the molecules that Jim and Saman have previously described. They're kind of an antisense molecule. So, and what it's aiming on doing is sort of preventing this thymidylate synthase from being made by the cancer cell, because the cancer cells will tend to use thymidylate synthase as a means to continue to proliferate and perform and get those tumors forming in the patients.
We don't want that.
And we don't want that.
Right.
So we're kind of thinking that with this new technology, with this new antisense technology, we can prevent the cancer cell from even making this thymidylate synthase that it might rely on.
In essence, then, Christine, what you're talking about is catching it right at the very beginning before it has a chance to grow and spread. And
either catching it right at the beginning or sort of taking away one of the ways that it's able to continue to sort of live propagate in patients. And then we'll use this in combination with chemotherapeutics that will hopefully help to catch anything that has been made, any sort of thymidylate synthase that has been made, and ideally we'd like to see, the tumor cells dying or the tumor cells no longer proliferating, so not getting any bigger in the cancer patients, and that would be a big goal. And another potential sort of interesting avenue to explore is that these antisense molecules can potentially help patients who have become resistant, as Jim mentioned, to chemotherapeutics, become more sensitive to them again. So sometimes they might make more and more of this TS molecule to sort of evade dying by chemotherapeutics that are targeting this TS molecule. So it decides, I think, well, the chemotherapeutic's trying to take this away from me, I'm just going to make more. But if we can prevent the cancer cells from making more, then we can potentially make these patients sensitive again to drugs at unharmful levels and catch their tumors and cancers before it continues to get out of control.
That all sounds great. I'm just wondering, while we're quickly running out of time here, where can the students go to find out more information about the fantastic research that you all are involved in? Where would you point them?
So we're both with the Department of Microbiology and Immunology, but a lot of departments all over the university will do cancer research or clinical research or just research in general. So each faculty's homepage is pretty good, and I think Jim is going to have a new website at some point in time up and running.
We were talking about this morning. My website is way out of date because I haven't had time to upgrade it. So
are you doing it yourself, Jim?
Well, I think they
we'll
said
we'll find somebody.
They said
if
I don't do it, they're going to do it for me, one way or the other. So if I want it written one particular way, I'm going to have to get on it. But one thing to do is to go to the website of the London Regional Cancer Program, and that's part of the London Health Sciences Centre. But just remember that, London Regional Cancer Program, and look up Cancer Research Careers and Training. And there you'll find the websites of all of the scientists, and there are about 17 of us. All of the scientists who are working in the Cancer Research Laboratory program, and there you'll see the work that we're doing in our laboratory. You'll see cancer imaging, which is a way of trying to detect cancers very early, because the earlier you can detect cancers before they develop some of these very nasty characteristics, the better chance you'll have of treating them. And then you'll see people talking about breast cancer research. You'll see people talking about head and neck cancer research. You'll see lung cancer research. You'll see all kinds of things going on there at each one of the individual laboratories. So please look up the London Regional Cancer Program website. Look up Cancer Research and Training. There you'll also find another website for the CART program, C-A-R-R-T, which stands for a very long name, which you shouldn't worry about. But it's a training program that Saman and Christine are both involved with, and that's a global research training program for cancer research in London, where we have many, many students working on all kinds of projects. And we get together to deal with the big problem of how we move new discoveries from the laboratory into the clinic. And how we do that by interacting with the industry, some of the private sector partners, some of our clinical partners, the people who are actually treating the cancer patients in the clinic, and people from many, many, many disciplines, sometimes even as far afield as exercise psychology and business management. All of these have aspects that are important to cancer research, and you'll find out about those aspects if you go to the CART website.
Well, I'm going to go look that up. I can't wait to find out what business management has to do with cancer research. By the way, we'll put all of those links at the TWISE.VROC.ca website. It's TWISE.VROC.ca. You can find all the links about Jim and his graduate students there. And Jim and Saman and Christine, thank you very much for coming on the show today. We really appreciate your time. And I know I learned something. I'm sure the students out there as well. That's another show for This Week in Science and Education. Catch us same time next week. Thanks a lot, everybody. Have a good week.
So long.